Our project was basically about generating molecules or other structures that have some conditions. And what is the overall problem? So overall, for example, when generating drugs, we would like to decrease the entire search space and generate drugs that satisfy some conditions. These conditions can be numerical, but can also be in a format of scaffolds or other substructures. But in our work, we focused on some numerical properties that molecules or peptides will satisfy. Yes. So in our work, what we did is generating molecules or peptides that have properties that are in some way computable. For molecules, these are deterministic properties that were computed using RDKit. And for peptides, these were properties that were computed by Oracle. So a quick introduction about the representation. What we want to do and leverage in our work is some representation of molecules or peptides in a string format. Smiles can be easily represented using the alphabet that consists of letters as well as some additional characters like equals. And peptides can be also represented by the alphabet, which helps us and allows us to use some natural language techniques.
All right, so what we wanted to check was to first generate some molecules or peptides de novo, so without any reference peptide or molecule. And the second thing was so-called prototype generation, so a setup in which we are basing our generation on some starting protein or molecule. So first I'd like to talk about the de novo generation and about our ablation study of different aspects of the transformer decoder-only architecture for de novo conditional generation. So as Asha said, we are dealing with numerical conditions. So these are just scalars. We have several of them, and we have to input them to the transformer model somehow. And we typically have some type of condition. So for instance, the molecular weight of a molecule and a specific value for this condition. So we have condition type and condition value, and we may think how to fuse them together, how to join them together. And here we checked several types of doing this. So first we did summing, which is the most trivial one. Second was doing some concatenation and projection. And finally, fusion by concatenation and then some MLP. Besides that, we've added so-called memory tokens or transistors, as they are called in some VR papers, which are additional tokens to be used for whatever reason by the transformer model. So we just appended them to the beginning before the conditions. And finally, we used these different additional tokens for auxiliary predictive tasks of the conditions which are used for the conditional generation. And here we are doing it in several setups. We are using CLS tokens for the sequence, or these memory tokens. And we are doing the prediction based on these CLS tokens, or we are doing unmasking of the condition values based on the condition type embeddings. So yeah, so we have several types of these predictive text. As I said, we may use the CLS token, which looks at the memory tokens of the sequence embeddings, or unmask the condition values from the tokens which have similar attention patterns. And well, one may ask why do so? Perhaps we may cause the model to store relevant information in places we have intuition may be important for the conditional generation. But it turns out it doesn't matter. But we checked also some additional things in this architecture. So some preprocessing of the conditions, what is the attention mask for the conditional prefix, and so on. And yeah, this brings us to the evaluation part of the de novo generation.
Yes, exactly. So we evaluated several settings that Przemek mentioned, as well as some baselines. And our main evaluation metrics were at first validity. So if the model produces valid smiles or peptides, uniqueness. So how often it generates the molecule that didn't occur before, as well as novelty. So are these generated molecules in a train set or not? We also compute mean average distance, which is very intuitive metric because it's averages over differences of the predicted and targeted conditions. So at first, before we present results for de novo generation, let me introduce you with one of our baselines. This baseline is called HEMFM, and it's a pre-trained large language model that is used for various tasks, for smile generation and other. And how pre-training of this model worked was very easy in terms of NLP techniques, because it just this model was learned to auto-regressively predict the next token, so the next element of the alphabets. And it was pre-trained on extremely large data set that consisted of known and at least to some extent like discovered smile strings. And for this particular task, so for conditional molecular generation, this model was fine-tuned by feeding a scaffold or the conditions in the as tokens that, and after the tokens there were provided either the characters of the scaffold or the MLP, so embedding of a given condition. So because the paper didn't consist of a broad, it didn't contain too many results, we conducted the evaluation on our own as well, on our datasets. And small spoiler, basically our models are almost are compared to this model, but they are significantly smaller. Yeah.
Yes, and here are the results for the ablations I've mentioned. And it may be seen that there are no significant differences visible besides the experiments which used positional embeddings for the conditional prefix. Here you can see how the marginal distributions of the conditions generated by our model match with the actual with the conditions from the reference datasets. And here are the results of the generation based on grid. So here we are specifying some grid of 4 conditions, and for each point in this grid generating a bunch of molecules, and here are the marginal distributions for each of the conditions. It's visible that they align. They're oscillating around the mean. Here are some example molecules which are novel and have a high drug likeness score, so potentially may be used as drugs. And now I'd like to move on to the different data type. We've talked about molecules so far. We did also we trained model on the peptides as well, and here we use pretty much the same model as before, but adapted it to be an autoencoder to have capability of first embedding some peptide and then sampling around it. So as I said, we took the pretty much the same model, but we used it both as decoder and encoder. In the encoding part, we only changed the attention pattern so that all it's all to all attention, and then extracted the pooled embedding of the peptide and obtained CLS token embedding, which was then passed through latent module, which allowed us to extract latent representation for this peptide. And this model was implemented in a manner which allows to both for both de novo generation and lead optimization. But we haven't investigated in detail how to train the model well to be good on both tasks simultaneously. So when it comes to latent models, we wanted to check several variational autoencoder types. So vanilla variational autoencoder, Wasserstein autoencoder, which uses some Wasserstein distance to compare prior and posterior, and finally importance weighted autoencoder, which theoretically helps to improve the band, which helps to leverage more samples in the latent space. We also check different posteriors and priors. And yeah, here is just some result for this for training with both objectives, just to show that training in this way may converge. But let's go to the results. Here are the results on the task of shifting marginal distribution of MIC, which is a condition for, which is a property of a peptide saying how good a peptide is as a drug targeting specific bacteria. And here we are comparing on a quantile plot the distributions of the reference dataset, so the datasets of these prototypes, which are then embedded to the latent space, and distribution of the new derived peptides which had shifted conditions of the originals. So we have 4 conditions. We are specifying shift vectors for each of the conditions and then generating a population of peptides. All right, and it like a hot take here is that if you add more conditions, then you may shift the distribution more. On the right, you can see that when we are shifting more conditions than the target one, we can actually shift the target one more. And this is likely due to the correlation between these MIC properties. All right, we also, besides this bug evaluation, we wanted to check whether we may improve the already known drugs. It turns out it's possible. For some of the expert-derived peptides, we can actually get better MIC, but for some other which are out of distribution, it's not possible. So we run out of the time, but just a final note, we also checked whether the peptides have drug-like properties via well-established procedures, and it turns out that these peptides are not very strange and potentially have drug-like properties. So thank you. We are ready to take any questions.
We don't have questions. So I have a question regarding this these Q2 plots. So
Sure.
here it was unconstrained, or
All right. So unconstrained in what means?
it's conditional, right? So you send the conditional. And you...
Yeah, we are adding. Whenever we have 0 in this shift, it means that we are padding this condition. So if we have some non-zero shift, it means that we are trying to improve on this condition. So in the first setup, we are changing just the first for the first bacteria, and this is for this first bacteria also. We may see that we are able to shift somehow the marginal distribution, and here like the same QQ plots, but extended with the shifts which include changes for other bacteria. And like surprisingly, we can shift more than at least for, but there is a thing, at least for the this part of the distribution, it's not visible here, but if one looks closely, then it might be seen that here these lines crosses. So we may shift more in this region of the distribution, adding these additional conditions, but not for the region of interest actually, because we would like the peptides with the lowest MIC to be used as potential prototypes in this generative setup.
Do you have any other questions? Then thank you once again.